So, welcome everyone. Thanks for joining in this week's ASAR Talk. Today we have Dr. Diwakar Kulkarni with us. He is a retired virologist and he's also the former director and principal scientist at ICAR-National Institute of High Security Animal Diseases in Bhopal. He has over 120 publications with 974 citations, and he is a recipient of several prestigious awards, including the recent Dhanvantari Award. Today, he's going to talk to us about the emergence of zoonotic viruses with reference to the socio-ecological factors. And this will be mostly about the Asian subcontinent. So thank you, Dr. Kulkarni, for accepting the invitation and for agreeing to talk to us today.
Good evening, and thank you for inviting me to deliver this talk. I have already committed, and I am thankful for the ASAR, or Association for Socially Applicable Research, for all these activities, and they are very encouraging and need-based in today's environment. My talk will be on emerging and re-emerging viral zoonoses. So let us start with the topic. Ivan Sherman has expressed a specific need for control of a disease. He says, control of a disease takes more than an understanding of biology of the pathogen. It requires proper financing, national will, and strategies for winning the public trust. Disease control also requires surveillance and projections of how the infectious agents will spread along with a consistent and equitable application of control measures by a robust healthcare system.
Dr. Kulkarni, sorry for interrupting.
Yeah.
Are you planning to share your screen?
Yeah. It has not been shared.
No, I don't think we can see it right now.
Okay.
Yeah, now we can see it. Thank you. Yeah.
So, sorry for the inconvenience. I'll start again. I will talk on emerging and re-emerging viral zoonoses. Am I audible? Am I clearly audible?
Yeah, I think there is a little bit disturbance in the background, but we can hear you. That's okay.
It's raining very heavily outside. That may be one of the reasons.
Yeah, it's okay. We can hear you. Yeah.
Erwin Sherman has communicated that control of a disease takes more than an understanding of the biology of the pathogen. It requires proper financing, national will, and strategies for winning the public trust. Disease control also requires surveillance and projections of how the infectious agents will spread along with a consistent and equitable application of control measures by a robust healthcare system. Now, when we talk of emerging zoonosis, it's a growing threat to global health. It is a cause of huge economic losses in the past 30 years. Many old infections are reappearing, they are spreading like Kyasanur forest disease. Nearly 2/3 of human infectious diseases arise from pathogens shared with animals. And in a recent 2-3 decades, almost 75% of emerging pathogens were of zoonotic importance. So you can imagine that whatever is coming as a new disease, it has something to do with animal. And I am a veterinarian, so I can tell you with more confidence that the problems are multifold. This underlines the need for multi-sectoral collaborations, including clinicians, public health scientists, workers, ecologists, veterinarians, and economists for effective management of prevention of zoonotic diseases. Now, why these particular zoonotic problems they arise? The underlying causes seem to be mediated by human action in most of the cases. We ourselves are responsible for that. So there are like changes in land use, extraction of natural resources, greed in animal production systems, trade and traffic, global warming, reduced plantation, cutting of trees, civil constructions, encroachments of forests, grabbing resources of wild animal food, diverting water resources from forest, killing snakes, improper garbage disposal, prey animal utilization by humans, unlawful charity for wild and peri-domestic animals, and so on. This last point, unlawful charity. Everybody wants to be wants to show himself as a charitable, and then we make the habits of the animals in such a way that they are more and more dependent on human life than the wildlife. There are various risk factors, which we call it as a predisposing factors, which may contribute to the emergence and spread of zoonotic disease agents, including social, like human behavior, mobility, demography, and public health aspects; technological, which production systems and medicine; ecological, animal management, agricultural practices, fisheries and environmental pollution and global warming; and microbial, wherein we get evolutionary changes, mutations, recombination and reassortment of genes, and so on. The public health burden of zoonosis includes outbreaks of viral pathogens such as Ebola virus, influenza A viruses, that is avian influenza and swine influenza, including bird flu and swine flu, Nipah viruses like newly appearing Nipah and Hendra virus, tick-borne viruses like Crimean-Congo haemorrhagic fever and Kyasanur forest disease, and coronaviruses, which we are facing now, starts from SARS, then Middle East respiratory syndrome coronavirus, and the current epidemic of SARS coronavirus 2, or what we call the COVID. In this presentation, because we have got a limitation of time, we will limit our talk to emergence of some important zoonotic viruses with reference to socio-ecological factors in Asian subcontinent, how different infections enter and spread in human population in India, highly pathogenic avian influenza, or what popularly we call it as a bird flu, H1N1 or pandemic swine flu, PDM 2009, Crimean-Congo haemorrhagic fever, what we popularly called as Congo fever, and Kyasanur forest disease or monkey fever. Then since last few years, we are also facing a nuisance from Nipah virus infection, and everybody knows about SARS-CoV-2 infection. That also will look in brief. We first look at highly pathogenic avian influenza or bird flu, very popularly called as bird flu. It has got several subtypes. However, H5 and H7 subtypes are deadly to birds and can infect humans and other animals that come in contact with infected birds. Highly pathogenic avian influenza was first discovered in humans in Hong Kong in 1997, in which 6 cases of human being died, and since then it has spread over almost all over the world. Currently, the virus is not known to spread easily via human to human contact. Still, experts worry that H5N1 may pose a risk of becoming a pandemic threat to human beings. Up till now, from 19 countries, more than 862 human infections with H5N1, highly pathogenic avian influenza or bird flu, have been noted, with 456 deaths, which have been reported to WHO from 19 countries. And there are several unreported figures. It also includes Canada. It also includes a case recently reported from India since November 2003. Indonesia, Vietnam, and Egypt have reported the highest number of human highly pathogenic avian influenza H5N1 cases to date. India has officially reported one case resulting in fatality. The boy from Punjab died in last month with bird flu. How this threat starts from bird flu? Highly concentrated poultry farming inducing stress, contact with infected birds, that particularly the migratory birds. And these migratory birds travel every year from sub-Saharan continent to several areas in deserts, Himalayas, and from several countries. They travel in search of food, and naturally these birds, they do not suffer themselves but carry the virus as a healthy carrier. When they come in contact with mixed farming in open, which is normally in developing countries, free range chicken, ducks, goose reared together, open air markets or live and wet markets. So birds are being sold there. Meat is also being sold at the same place in open environment without any biosafety measures. Non-scientific slaughterhouses of poultry, absence of adequate biosecurity in organized farms. So these are all factors which create big problems. And this is what is, you can see here, the migratory birds particularly carry the virus. They normally do not suffer from infections, and they carry very severe type of virus load in them. They travel all over the world, and some of them may become sick, some may just excrete the virus, and they travel through their migratory pathways. They shed virus in between, and when they come in contact with local birds, particularly ducks and poultry, which are free range or in open space, they infect them, they kill them, and then there are chances that it may pass on to pigs, other mammals, human beings, and then human may suffer. So this is the cycle of say bird flu, which creates many problems many times. See, this is a bird flu outbreak. You can see there is almost 90% mortality after bird flu, and all the birds will die. So daily in a flock of 1,000, you will find 200, 300 birds are dying, and within 3 or 4 days, the whole lot is dead. So this is a very serious as regards the poultry economics, but it is also creating threat, equal threat for human life. This is another picture. See, the biosafety gadgets, you can look at this picture. The biosafety gadgets, the team which culls the poultry or which kills all the birds in the vicinity to remove the host for the virus is with full gadgets. They have a biosafety suits, but the population here, without any protection, they are onlookers. And naturally, it is a very highly risky business to be in the area wherein the virus is openly moving. So this is a very typical problem in most of the developing countries. This is another problem. You can see here, the dead birds are thrown just near the water flow. The water is flowing, and part of the dead birds is also mixed with water. So naturally what happens is the whole lot of water is contaminated. It may pass on to several areas. It may pass on that infection probably in a very highly unscientific, risky manner. This is nowadays the peridomestic animals, birds like crows, they also create problems. They pick up the infection by picking up the dead birds, and naturally they also suffer. More or less, they are also responsible for transfer of infection from one place to other place. Now, this is another virus which we popularly known as swine flu. It is not a misnomer. Actually, swine flu is a misnomer. It is not exactly swine flu. It is H1N1 pandemic 2009 and originally started from Mexico. The new triple reassortant virus containing genes from human seasonal influenza, that is H1N1 human infection, H3N2 human infection, classical swine influenza, H1N1 virus, and H1N1 generated from Eurasian avian-like swine influenza. So all these three viruses or three groups of viruses contribute the genes to form a new virus, and this is what a pandemic 2009 H1N1 virus. While Eurasian avian-like and triple reassortant SIVs are the direct ancestor of H1N1 pandemic 2009, first time detected in Mexico. The classic SIV facilitated the spectrum of influenza A diversity in pig population during reassortant events from 1998 to 2009, and now it has become a regular feature in many of the countries. The H1N1 pandemic 2009 virus is the net result of concentrated pig populations being reared in close association of human, wherein lineages of mixed AIVs, SIVs, and human endemic viruses. I'm sorry, it is
Yeah.
Okay.
Sorry about that. I just in general, it would be great if people mute themselves after joining. Yeah.
Sometimes unmuting.
Yeah. Sorry about that.
In India, in 2000, but the factor is once adapted to human population, the virus doesn't require any animal intermediary, and the transmission from human to human is going on, and probably that has caused the pandemic. In India, in 2009 and 10, the pandemic resulted in 47,840 human cases and around 2,700 deaths. Since then, these swine influenza outbreaks continue to be reported every year, especially in winter. However, since 2017, the virus is spreading rapidly and showing epidemiological characters different from previous years. So we are getting a changed view. And incidentally, because of Corona, the medical attention towards H1N1 swine influenza has been diverted, and people are not much looking at that aspect of epidemiological characteristic changes. Look at this particular, a poor lady just feeding the pig without any protection, and this is okay for one pig, but you can look at this another picture. There is a piggery one side, the other side is a duck, and the lady is there. So human, duck, and pig, all three staying together, sharing several type of environmental factors, may create problems and may the virus, which is the influenza virus, which is a segmented virus, may take advantage of this particular situation. Then another serious problem is Crimean-Congo hemorrhagic fever. We call it the CCHF or Congo fever. It is a high-risk disease caused by a tick-borne virus of a Buniaviridae family. The Crimean-Congo hemorrhagic fever virus causes several viral hemorrhagic fever outbreaks with a case fatality rate of 40% or more. It is endemic in Africa, the Balkans, the Middle East, and Asian countries, particularly from Iran, Middle East to India. It is now getting itself established. In Pakistan, it is going on since last 50 years. But in India also, it has started appearing since 2011, and now almost every alternate year we get some of the outbreaks. The host are cattle, sheep, and goat. There is no apparent disease in these animals. So it is very typical of these viruses that they do not cause any clinical picture in any of the animals. They carry the virus, they transmit the virus, but they do not suffer themselves. And that is the reason wherein a clinical picture is not available. You have to show the detection of virus by some other method. The ostrich is also susceptible. An outbreak occurred at an ostrich abattoir in South Africa. Animals become infected by the bite of infected ticks. Principal vector is a hyeloma. And this hyeloma is a hard tick. The virus remains in the bloodstream for about 1 week post-infection, allowing the tick-animal tick cycle to continue. The virus is transmitted either by tick bites, through contact with infected animal blood or tissues during and immediately after slaughter. The majority of the cases occur in people involved in the livestock industry, disease, such as agriculture workers, slaughterhouse workers, and veterinarians. Human-to-human transmission can occur resulting from close contact with blood, secretions, organ or body fluid of infected persons, as well as clothes or bed linen of the patients. Hospital-acquired infections are more common in the initial stages as the confirmatory diagnosis takes longer time. So normally, for CCHF, presently the samples are only being sent to National Institute of Virology, Pune, from all over India. And it gives quick results, but still it takes 2 days at least to get the result, to sample to reach to laboratory and to get the result. And by that time, already the patient is hospitalized, and it is likely that the hospital-acquired infections pass on to some other patients or to medical staff or to paramedical staff. The medical nursing staff and patients' family members are more likely to get infected. The mortality rate from CCHF may be 30% or more, with death occurring in the second week of illness. In India, the disease has been reported since 2011 in Gujarat and then in Rajasthan, and the nationwide sero surveys have indicated a widespread prevalence of antibodies in almost all major states. So it is a matter of worry that we are getting, say, mortality also in humans, as well as the clinical form of this disease in human beings without any apparent clinical disease in animals. This is a hard tick hyeloma, and which is responsible for transmission of Crimean-Congo hemorrhagic fever. So this is a biological life cycle of the tick. It is transstadial during the developmental stages, various growth factors, the nymphs, the eggs, the larvae, and adults, they carry the virus among themselves, and naturally they pass it to livestock, they pass it to birds, they pass it to human, and then small vertebrates also. So all these factors, they spread the disease. The only figure is animals do not suffer, but human being suffers and sometimes succumbs to death. Tests on patient samples present an extreme biohazard risk and should only be conducted under maximum biological containment conditions. That is biosafety level 4 level laboratories only. Tick control with herbicides like chemicals intended to kill ticks is the only realistic option for well-managed livestock, because in a village population where animals live besides human beings, the finding of tick or the biting of tick on the owner or the worker is a very common practice. So they don't bother. They simply remove that tick. That's all. But the tick might have carried the virus, and that might have transmitted the virus to human being by that time. the only way to reduce infection in people is by raising public health awareness of the risk factors, reducing the risk factors of ticks to animals to human, animal to human, and human to human. So all these ways of transmission in the community, and educating people about the measures they can take to reduce exposure of the virus. Healthcare workers caring for patients with suspected or confirmed Crimean-Congo hemorrhagic fever or handling specimens from them should implement standard biosafety precautions. And this is very important. Unfortunately, our medical staff, or particularly paramedical staff, is not well trained in biosafety and biosecurity precautions. Another disease is Kyasanur forest disease, a tick-borne viral hemorrhagic fever endemic to southwestern part of India and is known for last 70 years. So this is actually coming under re-emerging infection. In recent years, the changed epidemiology has suggested that KFD may be considered as an emerging tropical disease in India. During 1957 to 2012, Kyasanur forest disease virus activity was limited to the Shimoga district of Karnataka. During 2012 to 20, confirmed cases are being reported from Tamil Nadu, Kerala, Goa, and Maharashtra. Even it has reached now up to Savantwadi, and it's being reported in many parts of Konkan area. Is caused by a flaviviridae member. Is transmitted to human through the bite of infected hard ticks. Here also, it is a hard tick, and this hard tick, particularly, it is not hyeloma. In this case, particularly, it is Haemophilus, and this is actually acting as a reservoir. Now, its clinical signs include fever, hemorrhagic and neurological features in human beings, and again, it has probably spread due to the movement of monkeys and small rodents harboring ticks, which act as a reservoir for the virus. Now, we are displacing monkeys from the forests because we are encroaching forest. Monkeys are coming to human population because of unlawful charity shown by human beings. And naturally, they are bringing out infections in the town. They carry the ticks. These ticks transmit virus to rodents. Rodents also carry the ticks. And again, this cycle goes on. They develop very low level of asymptomatic viral load in their system, which is sufficient to transmit infection to vector tick during blood meals. Earlier, serosurveillance studies have reported positivity in Gujarat, West Bengal, and Andaman Nicobar Islands. So, this particular southwest problem is likely to spread if we do not control it, if we do not care for this particular infection. You can see here, the monkeys, rodents, shrews are common host of KFDV. It passes on to rats. Rats come near human population, unsafe contact with infected animals. This tick cycle goes on continuously. The ticks are being harbored by cattle and other animals. These ticks may pass on to human beings and farmers and other things which are moving nearby the boundaries of forest. So all these pictures are the reason for worry for human beings. Now, a very serious infection called as Nipah virus. We'll look at that. It is currently going on in Kerala. Last week, we got a case in Kerala. It's limited to only few countries in the world. It's only Malaysia, Bangladesh, India, the three major countries which suffered from Nipah. In 1998-99, it was in a fruit bat, pig and human being in Malaysia. One accidental case from Malaysia to Singapore in 1999, which was few pigs were smuggled and it passed on to infection to human being. How is not exactly known, but those bats carrying virus were transmitted, they were transferred to Bangladesh and India in 2001, And since 2001, Bangladesh is continuously facing outbreaks every year. Now, same year, 2001, India also had one outbreak in in human beings, and then it was transmitted. 2007 was another outbreak in West Bengal, Nadia district, which was transmitted from bat to human. 2014, there was one outbreak reported from Philippines, which had a bat-horse-human connection. And from 2018, 2019, and 2021, it is from bat to human directly in Kerala, and there were a series of outbreaks in those three districts. In Malaysia, there were 265 cases and 109 deaths. In Singapore, there were 11 cases and 1 death. In India, 2001, we have a very big outbreak in Siliguri, which has 66 cases and 45 deaths. Bangladesh has got every year outbreaks from 2001 to 21, and in these 20 years, they have more than 321 cases and 225 deaths, around 70% of mortality. Then in India, in 2007, West Bengal, again, a Krishnanagar district, in India, Bangladesh border, we have got one family infected, and all of them, they died from that family. 2014, Philippines, the infection appeared via horses, and 17 human beings were infected, 9 of them died. In India, in Kerala, Kozhikode district, during 2018, it was a nosocomial outbreak wherein 23 people suffered, 21 of them died with 91% mortality. In 2019, there were, in Ernakulam district, there was only one case, and that was probably saved. It didn't die. But again, 2021, this year, last week, on 5th of September, we have one case, and naturally, this has died, the boy, 12-year-old boy, and fortunately, the infection was not spread from this particular place in Mallapuram district in Kerala. Pteropus bats, fruit-eating species, it's a larger species, popularly known as flying foxes, are supposed to be the natural host of the virus. They carry several viruses, including Nipah virus, without exhibiting the clinical signs, but excrete the virus intermittently. The bats also carry Ebola, but fortunately, we have not yet recorded any Ebola case in Asian subcontinent. This is the Pteropus giganteus colony. This photograph was taken by me from Malda district in West Bengal. And they just go on hanging themselves for a whole day and move in the night in search of food. The range is India, Pakistan, Nepal, Sikkim, Bhutan, Myanmar, Sri Lanka, Bangladesh, and Maldives. This bat is active mostly in the evening and at night. Indian flying foxes fly up to 20 kilometers for feeding. It roosts primarily on trees in a very large number of colonies. Almost in one colony, you'll find 800 to 1,000 individual bats, and they stay with the family. They have their own females, they have their own offsprings, and they move in families. Apart from fruit bat as a natural host, human, swine, cattle, goat, cat, and dog, as well as horses, can be infected. Only pigs were primarily infected in initial Nipah outbreaks in Malaysia before transmitting the infection to humans. It was later revealed that other animals such as dog, cat, and horses could also be infected when they come in contact with infected pigs. Experimentally, cat, golden hamsters, guinea pig, African green monkeys, and squirrel monkey can be infected. transmission of Nipah virus to human during this outbreak appears to have been bimodal. In Malaysia and Singapore, contact with pig was associated with infection in humans. The disease transmission in Bangladesh and India is different and without involvement of pigs. We have not recorded any incidents, either clinical or subclinical, in pigs. In Bangladesh and in some part of West Bengal, fresh uncooked date palm sap is consumed by a human being as a popular delicacy drink, and this drink is probably responsible for transmission of the virus. I'll show you the story. This is a very interesting thing to know how ecologically the factors move, and then we have to suffer. Since these collecting pots are mostly open and are easily accessible to fruit bats, the sap gets contaminated with the virus-laden saliva, and other excretions of bat and NIV is transmitted through persons drinking this contaminated sap. Contact with excretions, secretions, or handling inanimate objects from infected persons results in human-to-human transmission. Nosocomial transmission has been clearly demonstrated in recent Indian outbreak. Other commonly observed cause in human was climbing trees, having bat colonies or contact with bats. The domesticated animals can be infected by contact with infected pigs. Experimental infections have also been established in cats by intranasal or oral inoculation, and transmission of cats via semen is also possible. In utero transmission has also been demonstrated. Now, this is a story wherein how Nipah appeared, because bats are harboring the virus since ages. But what has happened exactly, that the virus appeared in human beings. The emergence of NIV infection or Nipah virus infection in Malaysia is believed to be due to series of complex interplay of ecological factors. The forests of Southeast Asia have been substantially reduced by deforestation for industrial plantation. Severe deforestation and industrial plantation over 2 decades from 1975 to 1995 in peninsular Malaysia, Sumatra, Indonesia have severely reduced the habitat of fruit bats, Tirapa species, the natural host of Nipah virus. And there was a burning of over 12 million acres of forest in Borneo and Sumatra in Indonesia in the fall of 1997. And that cast an extreme haze over Southeast Asia for months that blocked sunlight, reducing the ability of trees to flower and bear fruits. Even now also, because of the pollution, you'll find that from China to Singapore, whole lot of sunlight is hampered, and naturally that affects the plantations for flowering and bearing fruits. This was exacerbated by a drought driven by the severe 1997-98 El Nino Southern Oscillation event. These events led to an acute reduction in the availability of flowering and fruiting forest trees for foraging by bats. What should they do? They won't get any type of food. They won't get any type of fruits and flowers, and they have to move from there. And naturally, they traveled from Malaysia to Bangladesh. This caused them to travel great distances in search of sustenance and culminated in unprecedented encroachment of flying foxes into cultivated fruit orchards. So naturally, at the same time, the Malaysian government had established some of the pig farms outside the population. During the same period, intensive pig farming practices with planting of fruit trees lured the bats into unprecedented encroachment into the pig farms that had nectar-bearing trees. After sucking the juice from the fruit, the bats spit out pulp and drop half-eaten fruits on the ground, delivering a lethal dose of virus-laden saliva to voracious pigs. The pigs, thus picking up the contaminated fruits, suffered with clinical, subclinical Nipah infection. Pigs acted as an amplifying host and transmitted the virus to human beings. After the demise of pig farmers, the leftover pigs were fire-sold by surviving family members and transported illegally to other pig farms in Malaysia, transmitting the virus to other pig farms. This is a picture. This is all this photograph because I was working on Nipah for a long time. I worked on Nipah for 6 years. You will find on a palm tree, a small earthen pots are tied up at the top. They make a small scratch and put a channel to collect the sap. You'll find here, whole of this thing is with ant because it is a sweet juice or sap, they call it as, and such open mouth earthen pots are tied up on the trees. See here, you can see here the ants, and the sap is collected. In one night, they can collect even 2 liters of sap, and that is a delicacy drink. It is not cooked, it is taken raw, like what we take tadi or nira and something like that. Then there is another issue of SARS-CoV-2 or COVID 2019. It causes infectious disease called as COVID-19 coronavirus disease 2019 in humans, which was first reported in Wuhan, China in December 2019, or maybe even before that, because we have got no real information about the episode which is occurred in Wuhan part of China. On 11th March 2020, there were around 118,000 cases in 114 countries with 4,300 deaths, and at that time WHO has declared COVID-19 as a pandemic. Today we have got 22 crores 51 lakhs and 20,000 cases and over 46 lakhs 38,000 deaths all over the world. So I'm not going much into the details of SARS corona because SARS coronavirus 2 is very commonly known now for everybody. Everybody knows it, and they have adopted also so many practices. There are 7 coronaviruses that can infect humans. One is alpha, the another alpha, one is beta, and there is another beta. So these 4 viruses, they are transmitted through bats, and most of them, they have either camel as an intermediate host, or some of the times the hosts are not known. SARS-CoV, that is beta coronavirus, causes severe acute respiratory syndrome, and the reservoir is rat. The transmission intermediary host is cattle, and it goes to human. It was appeared in 2002, SARS coronavirus, and within 1 year, fortunately, it has disappeared from the global scene. Now there are not many cases of SARS coronavirus, but one has received a caution for this particular virus. Then there is a Middle East coronavirus and Middle East respiratory syndrome coronavirus, which was initiated in Middle East, and the virus again is responsible for bat, and it travels through camel. Now SARS coronavirus, novel beta coronavirus, or what we call a coronavirus 2019 or COVID-2, is a recent addition to this particular group. So these 7 coronaviruses, they affect human beings. Since 2005, dozens of new coronavirus have been isolated from bats, and there is evidence that human respiratory coronavirus, SARS and MERS, may each have originally emerged from ancestral bat viruses. As early cases of COVID-19 were linked to Wuhan market, it is possible that animal source was present at this location, either naturally or we can say artificially. There is a lot of controversy over the origin of coronavirus from Wuhan or China. A coronavirus isolate sampled from Rhinolopus affinis bat is almost 96% identical. It's genotypically 96% identical to SARS coronavirus 2. This is the Rhinolopus affinis, or popularly known as Indian horseshoe bat. Now, in case of Nipah, they are fruitivorous bats. They eat fruits, but these are insectivorous bats again, and they are smaller in size as compared to fruit bats. This is a big colony you can see here. They stay in hundreds and move with the small insects and other materials as their food. This is actually the animal market in Wuhan. This photograph is actually taken while there was an official order to close down the market of Wuhan. So the officers are there, which are making them the dealer to close down the market. But you can see here, the rabbits are there, the other animals are there, the bats, a large number of bats, they are selling for meat purpose, and lot of things which are illegal things, the hide, the skin, and so many things, they are being sold in open market. And that is the reason that Wuhan was the source for coronavirus. Now, if we look at public health aspect in India, the dairy farms, the poultry farms, the sheep and goat units all have unhygienic atmosphere. We are making our atmosphere or our environment prone for infections, and we are not looking at the hygienic control measures for avoiding the spread of viral infections. This is another public health aspect. We have very lovely pets with us, and we allow these pets to come too close to human beings. Are we ready to learn the lesson? What is the remedy? Now, Corona has given us the lesson. No crowding at any place. Mask, goggles, and social distancing is the key. No free roaming, no open eatables, no uncooked food. Wash unpeelable fruits thoroughly. Soak vegetables in clean water thrice if taken raw. Wash vegetable once before cooking. No hugs, no shake hands. Wash hands with ordinary soap and ample water frequently. No need to use disinfectant like Dettol, Savlon for a common man. So this is actually called for advertisement that you use Dettol. There is no need. You use ordinary soap, but with ample water. Avoid removing mask and touching your mouth, nose, eyes on road, and no self-medication. If you follow these things, probably we may be able to control the things in a proper way. Otherwise, even our own relatives may not come to our rescue even at the time of death. So this is a case of a Nipah, wherein all the personal belongings and everything was disposed of by the hospital staff without any relative. Thank you for a patient listening. Thank you very much. You can ask any questions if you have, or you can refer those questions to me by mail also.
Yeah. Thank you, Dr. Kulkarni, for a very detailed and meticulous talk for our audience today.
Yes.
And I think we definitely, at least speaking for myself, I think definitely I learned a lot. And like one of the most important things that I think I learned was that we are seeing COVID right now, and we see that as our biggest problem right now. But this has a long and large history of ecological disruption and interacting with our ecosystem in adverse ways that has led us to this moment. So thank you for covering that in great detail. We have one question in the chat box right now from Kiran.
Yeah.
She's asking, amid the reports and claims saying that COVID is a biological weapon, is saying it's zoonotically correct?
See, any type of weapon, even if it is a biological weapon, if you say incrimination of anything as a biological weapon is easy. You have to prove that, okay, this has been particularly used. Like COVID case, if you take example of coronavirus 2, there was yes and no for everything. And ultimately, the country's sovereignty will not allow you to disclose anything. You may infer yourself, okay, this was a biological weapon. Now, biological weapon during even World War or First War, at the end of First World War, was also there. They put blankets infected with plague into the territory of other country. Now, if you say like that, or even a smallpox virus was used as a biological weapon. Nipah can be used as a biological weapon. If you infect bats, a colony of bats with Nipah virus in laboratory and throw it out in any of the countries, they are going to create problem. But then confirmatively saying that, okay, this has been used by biological weapon by a particular country, it has got political ramifications, and probably it has got no technical relevance in that way.
Yeah, thank you. I hope, Kiran, that answers your question. I had, till we have few more questions, I quickly wanted to ask one question. So there is an increased focus on public health due to COVID. But how much of that focus, and especially like political movement towards dealing with these problems, is geared up towards zoonoses? Is that a political priority?
Yeah, very, very pertinent question. And it is correct pointing out. My first statement that therefore was that dealing with any of the infection, one has to have a political willpower. A country doesn't have political willpower, technocrats cannot do anything. You have so many doctors, you have so many hospitals, you have so many other things, but it requires funding. And what type of, say, resources are required? They are to be provided by political outfit. So for any country, you have to pressurize them. So naturally, you pressurize them for so many things, but you have to pressurize them also for public health aspect, that you provide us this particular thing, you prevent us from doing this particular thing, then only it is possible. Now, for example, some countries have now prevented government servants from entering into office if they are not vaccinated. Can we not do it very simply? In India also, we have got a large number of population which has not been vaccinated or which are not intentionally taking vaccines. Can we make them by compulsion that, okay, if you want this particular facility, you have to get yourself vaccinated? This has to be done by administrators or the politicians, and ordinary man cannot do anything. So, like, it doesn't require any law, specific law, but you can make the people to follow particular rules. Like now, you'll find out in next 3 or 4 years, our tuberculosis proportion will be reduced. Why? Because we have stopped spitting on the road. We have definitely reduced tobacco consumption. Why? Because there was lockdown, there were no shops were available, whatever. The reasons may be multifarious. But ultimately, if we say that this is particularly has to be done, administrators have to be pressurized for that. Otherwise, they will not do it. It is sure. No administrator would like to do unpleasant thing for public. And this is anything you say about public health, it is unpleasant. If I am dying only, I will listen. If I am not dying, I am healthy, I will not listen to anybody. So this is a political willpower. Surely that is there. It is a very pertinent question you raised.
Yeah, thank you. Do we have any more questions? You can just go ahead, unmute yourself and ask your question. Okay. If not, I think we will.
Yeah, you can forward those questions to me. I'll reply on mail also. No problem.
Yeah, we'll do that. Yeah. I mean, you can see Dr. Kulkarni's email on the screen right now. I will also type out the ASAR email ID if you want to send your questions there after the talk. and we'll forward those to Dr. Kulkarni. But thank you everyone for joining. Thank you, Dr. Kulkarni, for such a wonderful talk. And we'll make the recording available to the participants sometime soon. Thank you so much.
Thank you, Siddesh, and thank you, Dr. Surabhi. She is ill, I suppose, so she couldn't join. But anyway, it's a good opportunity to have interactions with you, and we'll be again meeting if you need something.
Definitely. Yeah.
Okay. Thank you.
Yeah. Thank you.
Shall I leave?
Yeah. Thank you, sir.
Okay.